Alright, ladies and gentlemen, this is going to be the next video on biology. I'm going to do a little bit more of a traditional slideshow reviewing DNA replication. Let's get started on this next video. Now, if we go through and we review DNA replication in eukaryotes, where is it going to go through and occur? The location is going to be in the nucleus. We're going to see that the quantity of DNA is going to be 1,000 times more than prokaryotes. If we go through and look at this, the method is what we went through and looked at in the last video. But the difference is it's going to go through and start in dozens to hundreds of places, and it's going to create hundreds of replication forks that are going to go through and form. So if we zoom in on this image here, we have our old and our new sets of DNA with the process that we went through and talked about. But what we can see is multiple sets of replication bubbles that are going along and going to join as replication goes through and occurs. Now, contrary to replication in prokaryotes is going to occur in the cytoplasm. It's going to come from a singular circular DNA molecule. Now, the difference here is it's going to usually start in a single place along that chromosome or along that section of DNA. So what we can see if we zoom in on the image here, we have one origin of replication, and that replication is then going to proceed in two different directions and move along that circular piece of DNA until it's gone through and been copied. So the main fundamental differences here, and it's because of the structure of the DNA when we compare prokaryotes and eukaryotic organisms. Now, what we want to do is we want to review the process of DNA replication in eukaryotes. So number one, two strands of DNA are going to be unzipped by helicase. The complementary strands are going to go through and be copied. And then we're going to see the DNA polymerase enzyme join individual nucleotides to form the new strands, which are also going to proofread for mismatches and errors. So we're going to go from one set of DNA, two separate strands of anti-parallel sets of DNA are going to be created, and eventually we're going to condense after replication this into a chromosome, which is then going to go through and be used for mitosis and meiosis. Now, I also want to go through and discuss two important scientists here. We have Watson and Crick. Watson and Crick discovered the molecular structure of DNA. They proposed DNA consisting of two spiral wound helical chains. This is in 1953. Now, they're really the first ones to go through and demonstrate how genetic information is passed through DNA in genes. But a more important figure here is Rosalind Franklin. Rosalind Franklin was not given the credit that she deserved, unfortunately, because she was a woman in a male-dominated field in the 1950s. Now, Rosalind Franklin's research with X-ray diffraction actually helps build the model that Watson and Crick end up using there. But again, she's not given the credit in any of the papers because she's a woman in that time period. But her important contribution to science is what led Watson and Crick to go through and come up with that double helix structure. Now, lastly, what we need to go through and discuss are telomeres along chromosomes. Now, a telomere are short repeated DNA sequences that prevent genes from being damaged. They're at the tips of the chromosome. You can kind of think of them like aglets, that little plastic piece along your shoelace. Now, there's an enzyme called telomerase, and it's responsible for helping make the telomere in these repeated segments of TAGGG. And again, what this is going through and doing is it's going to go through and prevent gene damage. Now, over time, as these repeated segments go through and occur, the telomere is going to be shortened. It's going to get shorter and shorter and shorter, which is going to go through and cause damage to that chromosome. Eventually, it's going to get so short that the telomeres no longer exist, and we start getting into coded segments of that chromosome. Because of this, what we can see is there's a limited amount of times that cells can go through and divide. In some cells, it's as many as 100. In some cells, it's as few as 3. We generally see about 50 cell divisions for normal somatic cells that we go through and look at. And it's because of these telomeres. As the chromosomes go through and age, the telomeres go through and shorten. Because of this, we see cells go through and eventually expire. Now, this is going to be the end of the video. There were a couple of things we want to make sure that we get out of this video. Did we review replication? Do we understand the difference between eukaryotic and prokaryotic replication? And then do we understand the fundamentals associated with telomeres? This is going to be the end of the video. We'll see you all in class tomorrow.